Different developments in next generation sequencing has led to various applications where we can apply these techniques. So we can say we are living in a kind of an omics era. So we have a lot of omics around us. Genomics, where we can do exome sequencing, where we can do whole genome sequencing. Then transcriptomics, where we can sequence RNA, and within RNA, if we are sequencing microRNAs, we can call it as mirnomics. Epigenomics is where we can sequence special modifications on the DNA, so that may be important for gene functions. So let's look into it, what these major areas are in detail. So here we see there is a cell that has a nucleus, and in this nucleus in the middle we have the DNA, and then this DNA has some modifications. So here we see there is some methylation on the DNA. DNA wraps over histones, which is right here. So any modifications on the DNA we study in epigenomics. If we study the sequence of DNA, it becomes genomics. Then DNA produces RNA. So if it is a messenger RNA, we can classify it as transcriptomics. And microRNAs are special RNAs that can somehow block those messenger RNAs, and then they can control the gene regulation. So that study is mirnomics. And then they have within them different areas or different branches. We can split them further. Here in this graph we see these bars where experimental costs are shown in red and information content is shown in green, whereas the clinical utility is shown in blue. So if we look into genomics, they have mostly the clinical utilities are where we have the targeted sequencing, then exomes, whole exomes, and if we go down and we do whole genomes, it's less applicable because it may be related with the cost. As far as the information content is concerned, obviously they are other way around. More information at the bottom, same way more cost at the bottom. So whole genome is an expensive task as compared to the targeted genome sequencing. We look into epigenomics. The trends are we can get more information at the bottom of these all four groups. So where we can have whole genome bisulfite sequencing, microRNA editing, that is more informative. So costs are similar if we look into them. Same way transcriptomics. To do that, the costs are same for all of them. Whereas if we do some specific tasks like RNA editing, where we study some changes in the RNA, so there we can have more information out of it. So mostly the work is done in this area in gene expression studies nowadays. There's a technique called as RNA-seq, which is highly used and under great pace of development. So we will talk about that in a later section. Whole genome sequencing involves sequencing of the whole genome. So genome is the total DNA complement of the cell. We can do sequencing. Then after we do sequencing, the next task is to put all those short sequencing reads together into what we call it as assembly project. And after we assemble these genomes, what we do next is we can identify the protein coding genes, and we can do some annotation works. And then in this way we can also study different variations present in that genome. So we can have a genome of one individual, and in comparative genomics what we do is we compare that genome with some other individual, so we can get the idea about variations among these two different individuals. In exome sequencing, we don't sequence the whole genome, but we pay attention towards those regions within the genome, even within the genes from which the expression is performed. We call them exons. So if we capture them and if we sequence them, we can target specific areas of the genome. So it is cost effective method which is mostly applied to study different genetic diseases. In transcriptomics, as we have seen earlier, we detect the transcriptome repertoire, that is the total RNA complement of the cell. So that can be a messenger RNA, ribosomal RNA, transfer RNA. We classify them as coding versus non-coding RNAs. So non-coding RNAs are everything except messenger RNAs. In mirnomics, we study microRNAs. So microRNAs are small fragments, normally 22 nucleotides in size. What they do is they are kind of complementary to the messenger RNA, so that they get attached to the messenger RNA and block its expression. So in this way they control gene regulation. Epigenomics is where we study everything except the genome or above the genome level. So we are looking into different modifications on the DNA. So that may be the modifications on the DNA nucleotides itself, kind of methylation, addition of methyl groups. We may be looking into the modifications on the histones. So they also have some important role in the development and some other genetic chromosome activations, etc. So far we have seen that NGS can be applied into genomics, transcriptomics, meronomics, and epigenomics.